of interactions. Long title.
Okay. Hello. Thank you. So I will talk about an approach for gene network inference based on epistasis analysis and using mutant data. So overall, I will first talk a bit about the epistasis analysis and the motivation behind our work. Then I'll present our factorized model and an approach for assembly of gene networks. Then some results on our performance, and finally I will conclude. So let's start our journey. And the journey can start with a simple toy example that is concerned with two genes, U and V, and a phenotype which is observing the fur on the nose tip of Heffalump. So this is this imaginary elephant from the Winnie the Pooh, those who remember. So let's say we have some data, and the data that we observe is what's the phenotype of this organism or this elephant when a certain gene is mutated, is knocked out, sorry. So the mutation in gene U results in the fur, in the phenotype which is shown as fur on the nose tip of Heffalumps. And then the second data point that we have is that mutation in gene V results in no fur on the nose tip of the elephant. And the mutation in double mutant also results in the no fur. So the question is, what can be a network and basically relationships between these genes and the phenotypes that we are interested in? And obviously, in normal, in our usual world, these organisms, these elephants, don't have the fur on their nose tips. So a relation that is consistent with this, our observation, is shown here on the right side. On the right side, and here we can see that gene V is the one that causes the fur phenotype, and gene U represses the gene V, meaning that a mutation in gene U will result in failure to repress gene V, and thus we will observe the fur phenotype, meaning that the model that we have, the simple model, is consistent with our observation. And this is the epistasis, which can capture the interaction between two genes. And the interaction, the idea here is that two genes interact if a mutation in one gene masks the effects of perturbation in another gene. And we described here only interaction between two genes and the epistatic interaction. So the idea of epistasis can be further extended to capture the logic of parallelism. Well, both genes have an effect on the phenotype, but they are not in epistatic relationship, or partial interdependence when one gene acts through a separate pathway. But this, again, is just relationships between pairs of genes. So our goal overall is to construct detailed multi-gene networks that are consistent with observed pairwise gene relationship data that we have. And in more detail, we would like to have an approach that can handle quantitative data, meaning quantitative phenotypes that we measure, such as the fitness of an organism or expression of a reporter gene. We would like to have an approach that can handle measurement noise and can easily handle data sets with thousands of genes. So for that, we proposed a factorized model that can address this problem. And the input to this factorized model consists of three parts. And these three parts are obviously generalization of our two example data that we had there. So we had, for a given set of genes, here we have just 4 genes in this cartoon. We have a vector which represents the phenotypes of single mutants. Then in number 2, we have a matrix where each entry is a phenotype of the corresponding double mutant. And finally, what we have is also an expected double mutant phenotypes. So this is the matrix where the entry UV encodes the expected phenotype of an organism if the corresponding genes would not interact. So now the question is, how do we get this data, especially this expected double mutant phenotypes. And there is quite some substantial amount of literature on that, how to estimate an expected double mutant phenotype. And usually there are 4 approaches to do that, using different functions such as minimum, the product, logarithm, or addition. And this depends on the phenotype that we measure. But this value is usually obtained through phenotypes of single mutants and in some way combined to get the idea of what would be the what would be expected phenotype if the genes would not interact. So this is the input part. This was the input part. So what we do next? So the next part is actually the factorized model. So here we have our input data matrix, the matrix of double mutant phenotypes, and we factorize this matrix into a product of 2 small lower dimensional matrices, here shown U as U and V. But the idea is that we do not use only a linear latent model, but we pass the dot product between these 2 matrices, which contain the latent feature profiles of our genes, through a parameterized logistic function for which parameters we learn during our inference procedure. And overall, our aim is to reconstruct the double mutant phenotype matrix as well as possible. So allow me only one slide that describes the idea behind the cartoon shown before, which is we basically model each observation of double mutant phenotype as being a sample from a Gaussian distribution, which is centered at our factorized parameter obtained by the product of corresponding latent vectors showing in the first row. And then we impose the Gaussian prior on the latent factors of matrices U and V and derive the posterior probability of the unknowns given our observed data, meaning the unknowns in our case are both these latent matrices UV plus Xi, which encodes our parameterization of logistic space, given the data that we have, which is in this case the double mutant phenotypes. What we do then is select factorized model by maximizing the log posterior of this probability, and we selected for that, we formulate an objective function, which is an equivalent representation to map estimate of this posterior probability, and then employ a optimization procedure to estimate both the latent data representation and logistic parameterization. So once we have this latent data representation, obviously the next step is to assemble the G network. And for assembly of G network, we first score all pairs of genes according to different type of relationships that we model. If you remember from the beginning, we modeled 4 types of relationships: epistasis, partial interdependence, and parallelism. The first approach, the first step in the G network assembly is to probabilistically score gene-gene relationships based on our latent data representation that we got in the first step. And for that, we use the rules that are shown here on the right. And the basic intuition behind these rules is that the probability of a certain network structure between 2 genes approaches 1 when the factorized parameter of these 2 genes is close to the expected phenotype of that particular network structure that we have. And it slowly vanishes when the expected phenotype that should be obtained under a given network structure is different from estimated factorized parameter. So this schema is sort of a generalization of a standard epistasis analysis framework where the genes and the phenotypes could be only strictly on or off because it provides a smooth transition between different levels of our certainty. So for all pairs of genes that we have in our model, we perform this probabilistic scoring of these 4 possible relationships that we have here. So the second step is assembly of these multi-gene networks based on the pairwise relationship scores that we got. And the idea is that we would like to have the gene network that is consistent with our observations from pairwise scoring. And the network is a weighted directed network where the weights and the direction of the edges in that network are determined by our probabilistic scores that we got in the previous step. And the nodes in this network are obviously the genes that we have in our model. So the most important part of that last step of our gene network inference is that we assign genes to different levels based on whether it is for each pair of genes, we consider whether we have more evidence that these two genes are in epistatic relationships, then they should also be assigned to different levels. Whether if we have enough evidence that these genes are in parallelistic or partial interdependence relationship, then should be assigned approximately the same level. And once we get this assignment of levels of genes, we perform approximate topological sort of this weighted directed graph, which reduces the number, which produces the ordering of genes in the pathway. But what we do next is we would like to have the minimal such a gene network that is consistent with our pairwise observations. In this case, this means that we eliminate certain edges. These edges that we eliminate are violating and redundant edges. So violating edge is defined as an edge where we have both evidence that for a pair of genes U and V, we have evidence that both U is upstream, acts upstream of V, and at the same time V acts upstream of U. So this is a sort of violation, which is shown on the left side. On the right side, we see an example of a redundant edge, because a redundant edge is an edge, for example, between U2 and V2, because there is evidence of some intermediate gene. In this case, so the redundant edge is edge between U2 and W2. Yes, this edge is redundant in this model because we have evidence of some intermediate gene that acts between these two genes, in this case, V2. So as a final step, we remove violating edges and redundant edges. And we do that such that we take into consideration our structure that we have from previous steps, this assignment of genes to levels, and we retain those edges that exist within certain levels or between levels and are directed downwards towards the phenotype node. Okay, so that would be an overall approach. So this is here's just a schema of an algorithm, and the algorithm basically involves two primary steps. And the first step is the inference of factorized model, where we infer gene latent feature vectors that capture the global structure of our phenotype space and learn the logistic parameterization, which is a nonlinear transformation from the latent space to the phenotype space. And the second step is the step of performing pairwise scoring of all pairs of genes that we have, and according to all 4 types of relationships that we model, and assembly of gene network that is consistent with our pairwise scores. So let me talk now about our experimental part. And before that, I would like to present few other algorithms, or just mention them that exist. So there are only a few algorithms that are general purpose algorithms that are able to construct gene networks based on epistatic data or mutant data that we have. And one of the earliest was GenPath algorithm, which included inference rules and inference algorithm that could handle different types of relationships, but only qualitative phenotypes. And these limitations were nicely bypassed by an approach by Bettel et al. In 2010, when they proposed a Bayesian approach to gene network inference from quantitative mutant data, which proceeds in a way that it started with random network and then it iteratively improved the network by performing small local structural changes to the evolving network. And the approach that we propose in this paper is an approach that we refer to as RET, and this approach is conceptually different from the previous approach of APN. In particular, it's different because it simultaneously factorizes the whole phenotype data set that we have, and it produces the gene network as an output in a single-step construction. So the data that we consider in our experiments are two data sets. And the first one is the data set by Jonikos from 2009. And this data set is a data set of 440 genes, and they measure the unfolded protein response levels, meaning the expression level of genes that are involved in endoplasmic reticulum. And what our goal will be is to reconstruct several known or partially known gene pathways on genes whose perturbations are measured in this data set, and we will compare what are our predictions with, and how do our predictions relate to the ground network. The second data set is a recent data set from a couple of months ago. It's a bit larger, and they measured a different phenotype. They measured the sizes of the colonies of yeast genes. And our goal will again be to compare the inferred and true networks and relationships between genes that are included in these data sets. So we do this through measuring area under the ROC curve, or AUC. So here, for example, you see just a cartoon of an inferred network and a known network on the right side. And we see that for each test pair of genes, we know that either one gene acts upstream of another or vice versa. So the rate predicts the probability of epistatic relationship between the genes that are in the test pair. And based on that, we can evaluate the performance of the model. So here we analyze the ability of RET to reconstruct a known and linked glycosylation pathway whose genes are members of the Jonikos data set. And here we can see that in the top row is the network that was inferred by RET, and in the bottom is shown the true network according to known relationships between genes in the literature. And what we can see here that RET captures quite well many functional interdependencies between pairs of genes. In particular, it detects that DI2 and ALK8 genes, which are shown on this upper parallel path in RET, are functionally dependent on most of the lower stream genes. And what is perhaps worth mentioning is that this CVH41 gene is an example which is for which many measured interactions in our input data are missing. But just because we simultaneously treat the whole data set of double phenotype data, we can still place it correctly, which is upstream in this pathway. Now the next part was, or the next experiment was to evaluate what is the quality when predicting the ordering of genes in cake pathways. So again, we relied on the data from Jonikos and the test data set that they proposed, and they considered their genes that participate in common cake pathways and constructed a positive data set based as pairs of genes where one gene acts upstream of another, and the negative examples were constructed as pairs of genes where both genes are involved in a certain pathway, but one is not upstream of another. So our question here is, what is the quality of predicting epistatic relationship? So for that, we compared our approach, which is shown here by the red curve, with the approach by Bettel, APN. So we can see here that the performance of the proposed approach is substantially higher, which indicates that there is perhaps some benefit of our way of modeling. So finally, here is an example of inferred gene network on a set of genes from Surma et al. Data set. And in particular, they were focused on various aspects of lipid biology. And here is one pathway, tail-anchored protein biogenesis pathway, where the solid edges in this network indicate the edges that were inferred by read. So this was the final network that was produced by the method. And the dotted edges in this figure here show the known functional interdependencies between genes. So what we can conclude based on this result is that RIT is able to capture known functional interdependencies between genes quite well. So to conclude, RIT is a conceptually new approach for gene network inference from quantitative mutant data. It implements probabilistic epistasis analysis and assembles multi-gene networks from pairwise relationships that are scored in the model. And the key components of our model are factorization and nonlinear transformation. And we have shown in the paper that both these two components of our approach are crucial for its good predictive performance. Finally, in the experimental part, we observed that RIT is able to accurately reconstruct several known and partially known gene pathways with the accuracy that is above that of state-of-the-art approaches such as APN, and is able to do that very quickly in a matter of few minutes. So finally, I would like to acknowledge my advisor, Blaž Zupan and Uroš Petrovic from University of Ljubljana and ISCB for providing me the travel fellowship to come here. Thank you.
Very interesting work and very nice presentation indeed. I have two questions. One is the pruning method that you talked about, and you said some of them are contradictory if something is both upstream and downstream. My question is, my first question is, how does it work when you have multiple pathways? In one of them, one gene is upstream of the other, but we have also additional pathways, which in those alternative ones, their like order of them are switched. Another one is, I know like this is like directed like set of edges that you predict, but I'm just curious, how does it compare to, if you compare this data set to the genome-wide data sets that we have, the genetic, well, positive genetic interactions in yeast? What are the genes that, what are the interactions that you predict that are missing there, or vice versa, that were there, but you find it to be false positive and you predicted that they shouldn't be there? How do they compare? Do you have any result on that?
Okay, so first, if I rephrase your first question was, for example, if there are feedback loops in our network.
Yes.
Yeah, so in this version of the approach, the short answer would be that we do not model the feedback loops in our network. So that would be the short version. We could, by simply replacing some of the steps or this or changing the edge elimination procedure, this could be done. So to answer the second question, the principal goal or the aim was to construct directed networks, because the direction of the edges in this network indicate what is the ordering of genes, meaning the order of action of genes in the pathway. So we would like to know which is that gene detects upstream of another gene in a certain pathway for measuring certain phenotype. So this is conceptually completely different from the approaches which construct mostly correlation networks, which observe, for example, given a given a data set of genetic interactions, there are many methods that compute certain correlation between genetic interaction profiles, and based on that construct undirected networks.
Okay.
So here was the conceptually this is different.
Yeah. But to rephrase the second question, if we ignore the direction of predicted edges in your method and compare them as edges, how much does it overlap with experimental methods that we use for predicting positive genetic interactions among genes?
So actually for that there is an experiment in the paper which I didn't show here. So we basically we didn't take into consideration the directions of the edges, but just we evaluate what is our performance in predicting alleviating genetic interactions, for example. So there we compared our approach with the approach by APN and some Gaussian processes approach and a few reductions of the method, and we concluded or the experiment shown that our performance is at least comparable, if not substantially better than these approaches. But I didn't present these results here, but they are in the paper.
Thank you very much.
Thank you.
Hi, very nice work. I was wondering if I understand correctly, the factorization part starts with some random initialization of the matrices.
Yes.
So potentially, if you run it twice, you could get two different results at the end, right?
Yes. So this is.
Did you investigate the stability, or how much the results you show are depend on the initialization?
Yes, we did that, and the results for that, basically the graphs are in the supplementary information of material of that paper. So what we did is we did experiments on first, what is the effect of noise in the data on our prediction, and second, was that experiment exactly that you mentioned. So if we construct so what is the level of influence of initialization of the latent matrices on the final prediction? And for both cases, the influence was very low because we have this, so showing that the rate is a robust approach, and this could be explained by that we use the latent data representation to perform pairwise scoring of pairwise probabilistic scoring, and it seems that in that step, this step sort of has some indicates that the method performs robustly. We have these results in supplementary material.
Okay. Thank you.